Hello, my name is Simon Heels. I'm based in London in the UK, and I work at University College London, Institute of Child Health, Great Ormond Street Children's Hospital and the National Hospital for Neurology and Neurosurgery, all based very closely together in London. So thank you for the opportunity to talk to you today. And I really want to focus on medium chain fatty acids. We're all aware of the medium chain triglyceride ketogenic diet. And the particular fatty acids that we've been working on over the years is C8, octanoic acid, and decanoic acid. And they have revealed some very interesting findings for us, because epilepsy is linked in some studies to a failure of energy metabolism. And in our group, when we hear about a failure of energy metabolism, we start to think about a loss of mitochondrial function. And indeed, there are many studies to suggest that epilepsy may be associated with a loss of mitochondrial function. So our early experiments that we did were on cells, cultured neuronal cells, SHSY5Y cells, and we quite simply looked at the effects of octanoic acid and decanoic acid on readouts in the sense of how mitochondria may be performing and how many mitochondria may be present in the cell. So looking at decanoic acid in particular, because this is the one that gave us the most exciting results. So using a concentration of around 250 micromolar, here I have the levels that are seen in the traditional ketogenic diet that you will see that are achieved in plasma. So we use 250 micromolar and expose the cells to that concentration. And we saw, when we delved down into the function of the mitochondrial electron transport chain, we looked at enzyme activity, and mitochondrial complex 1 was significantly increased in activity. Complex 2,3 was not affected, and complex 4 was not affected. So already we've seen something, an increase in, perhaps some may argue, the rate limiting enzyme of the mitochondrial electron transport chain complex 1 was increased when the cells were exposed to a sensible level of decanoic acid. When octanoic acid was used, we found absolutely no effect whatsoever. So that fatty acid, no effect at all. If we move away from biochemistry, which we have to do sometimes, and look at electron microscopy, our colleagues in the histopathology department looked at those cells that have been exposed to decanoic acid and looked at the mitochondria. And here you can see the control cells, Vilca only. And here you can see the effect of the decanoic acid on the cells. You can see many more electron-dense mitochondria following exposure to the decanoic acid. So the biochemical level at the respiratory chain and at the electron microscopy level, decanoic acid is doing something to mitochondrial appearance and also to mitochondrial respiratory chain activity. We started to look deeper and deeper, and we started to see mitochondrial DNA itself is increased in content when the cells are exposed to decanoic acid. So that's a very important finding as well. We've done other things. We've looked at mitochondrial membrane potential, and that is increased when the decanoic acid levels are at the level of 250 micromolar. So when the cells are exposed to the decanoic acid, mitochondrial DNA increases, and also there's an increase in the mitochondrial membrane potential. So that became quite fascinating to us and starts to enabled us to postulate a mechanism whereby decanoic acid may be having a beneficial effect in the ketogenic diet. So then we wondered how it might start to build up in the brain. And so we started to do a head-to-head experiment with octanoic acid versus decanoic acid. And we found that octanoic acid was superior with regards to its rate of oxidation compared to decanoic acid. And that's because later studies showed us that the decanoic acid needs the carnitine shuttle to get into the mitochondria to be oxidized, whereas that octanoic acid does not. The brain does not have so much of the enzymes that make up the carnitine shuttle, and so it's a possibility that when decanoic acid gets into the brain, it's relatively inferior with regards to its rate of oxidation, and therefore it may start to build up. And interestingly, if you start to add a little bit of octanoic acid into the mix, it starts to even further protect the decanoic acid. So a mixture of around 80% decanoic acid, 20% octanoic acid gives you the optimum ability, we think, to increase the levels of decanoic acid in the brain. So decanoic acid seems to be the fatty acid of interest, but it's helped by octanoic acid, positive effects on the respiratory chain, and on the membrane potential and on the mitochondrial DNA. And we've extended these studies into other cell types and to replicate these findings. And now we've done some animal studies as well that are beginning to show similar findings. So it's all very, very exciting. How does decanoic acid work? We think it's through PPAR gamma, when it's acting as an agonist on PPAR gamma, and that's causing the positive effects we're seeing on the mitochondrial function I've just described. So what are the next steps? Well, this work has gone extremely well, and I'm pleased to say we've started to enter into a tolerability trial on adults and children, and this is a summary of the data we have found to date. So it's an early stage trial. So we've used a formulation in partnership with our funding body, VitaFlow, who have made a formulation. We've used a formulation with a high proportion of decanoic acid. We found elevation in the plasma levels of decanoic acid, but without a corresponding rise in ketones. So it's not a ketogenic diet. It's a decanoic acid elevating diet. We've looked at 35 children and 26 adults. 85% of the participants had previously tried or failed or unable to start a ketogenic diet. 66% of the children, 69% of the adults completed the study. The most common reason for withdrawal was GI side effects. The mean reduction of seizures was 50%, and only 3 children and 1 adult became ketotic. So it was not a ketogenic diet, as I mentioned earlier, largely. Seizure reduction correlated with the decanoic acid and the octanoic acid C10, C8 levels. So that's where we are today. Obviously, there's progression, but we are actually moving already from the laboratory data, from the laboratory bench into the patient already, but there's clearly a number of steps that we're involved in now. So thank you very much for your attention. And these are my collaborators who I work with on this aspect of this study. Thank you very much.